Hello students, you are welcome to SS7 of this course NSC 208. In this study session, we are going to have discussion on inflammatory response and chemical mediator. Although injury starts the inflammatory response, chemical factors released upon this stimulation brings about the vascular and cellular changes. The chemicals originate primarily from blood plasma, white blood cells, platelets, mast cells, endothelial cells lining the blood vessels, and damaged tissue cells. In this session, I'm going to introduce you to the biochemical mediators released during inflammation, plasma protein synthesis systems, chemotactic factors, and acute phase reactants. When you have studied this session, you should be able to describe the biochemical mediators released during inflammation. You should be able to highlight and explain various plasma protein systems, and then explain chemotaxis and its type. Now looking at inflammatory mediators. These are soluble, diffusible molecules that act locally and systematically at the site of tissue damage and infection, and at more distant sites. The effects of this inflammatory response are initiated by a complex series of interactions involving several chemical mediators, whose interactions are still only partially understood. Some of these are derived from the invading organism. Some are released by damaged tissues. Some are generated by several plasma enzyme systems. Some are products of various white blood cells participating in the inflammatory response. Biochemical mediators released during inflammation intensify and propagate the inflammatory response. Inflammatory mediators can be divided into exogenous and endogenous mediators. The effects in this response are initiated by a complex series of interactions involving several chemical mediators, whose interactions are still only partially understood. So, for example, endotoxin can trigger complement activation, resulting in formation of anaphylatoxin C3A and C5A, which cause vasodilation and increase vascular permeability. I talk of endogenous mediator. Those ones are produced from within the cell, which are innate and adaptive immunity, and they can be derived from molecules that are normally present in the plasma in an inactive form. So, we look at plasma protein systems. This contains four interrelated systems of protein that generate various mediators of inflammation. They are complement, kinase, coagulation factor, and fibrinolytic system. I talk of this protein system, they are interrelated and they generate various mediators of inflammation. We talk of chemotactic factors. This term refers to movement of leukocytes or cells in general induced by a chemotactic stimulus. Substances possessing chemotactic activity are called chemotactic factors, chemotaxins or chemoattractants. Besides chemotaxis, simulated directed migration leukocytes also possess two other types of movement. We have random migration, which is undirected spontaneous migration, and we have chemotactic stimulus, is provided by substances that can either attract or repulse the cell. Thus, chemotactic cell movement can be either positive or negative. The cell may move towards the source of chemotactic substances, towards an increasing polarization gradient or opposite direction. The positive movement is typical for leukocytes. Leukocyte chemotaxis is mainly responsible for the mobilization at the inflammatory state. Both endogenous and exogenous chemoattractants participate in this effect. Endogenous chemotaxis include bacterial oligopeptides of the FMLP type, lectins, the natural protein, some lipid and liposaccharides. Endogenous chemotaxins are produced by the host organism and are of humoral or complement factor or cellular type from different cells, chemotactics, cytokines, etc. So what we are saying necessarily is that this movement of leukocytes induced by chemostatic stimulus possessing this activity are called chemotactic factors, and they could be either attractive or repulsive. They either move in positive way toward the cell or they are repulsed from the cells. So these are what they call chemotaxin. So what we have discussed in this section is that we have recapped our knowledge of inflammatory process, and we have talked of the inflammatory mediators, which we said that some of them are derived from the invading microorganisms, while some are released by the damaged tissue, and some are generated by several plasma enzyme systems. Some are products of white blood cells who are come to participate in the struggle at the inflammatory site. This mediator released during inflammation intensify propagate the inflammatory response, and that response is now seen as inflammatory response in the host tissue. And we have discussed the plasma protein system, which involve kinase. These are chemical substances produced in response to inflammation. And we have complement system, which are produced as a defense mechanism for specific activities on the invading microbes. We talk of coagulating factors, which becomes a soldier and immobilize further movement of antigens, and the microbial antigens and the white cells that have destroyed them. They are localized in an area, not allowing them to move beyond that area, and they are called coagulation factor. They remain stagnant in that area of infection. They didn't allow it to come into the body system and generate into systemic infection. And then we have clotting factor. The clotting factor system is a fibrinolytic factor, and that one, what it does is that when coagulation is formed already, this system will just come in to break the coagulation, because at that time all the natural cells have been ingested by phagocytes, and with that they will make sure that the system does not suffer from that infection in systemic manner. And I discussed also chemotactic factors, which we said they are two in nature. The first one is that they may be attractive to the cell or repulse the cell. If they are attracted to the cell, they are called positive chemotactic cell, whereas those are repulsive of the cell are called negative chemotactic cells. And with this, we've come to the end of this discussion in this study session, whereby we have discussed on inflammatory response and chemical mediators. We have seen how the body responds to inflammatory system, and we have seen how chemicals are secreted and the effect of this chemical on the body. Students, we have come to the end of SS7 of this course, NIC 208, where we are discussing on cellular pathology. We have discussed on this session response of the body to inflammatory reaction, as well as production of chemical mediators, which will signal a lot of significant clinical manifestation of that inflammatory agent on the body. Until when we meet in the next study session to discuss something more interesting on cellular pathology. I want to thank you for listening, and I say best of luck.